Что?
Я включил дублирование экрана.
Оно работает, да? Я не слышу, что он говорит. Да, иди ко мне.
Стас, а мы можем на одном компьютере запустить, а с другого...
Уважаемые коллеги, доброе утро. Мы рады всех приветствовать на нашей очередной третьей международной научно-практической конференции «Гармонизация подходов к фармацевтической разработке». Разрешите слово представить первому проректору по научной деятельности Российского университета дружбы народов Костину Андрею Александровичу. Он является профессором РАН и член-корреспондентом РАН. Андрей Александрович, вам слово.
Добрый день, глубоко уважаемые коллеги. Я искренне рад, что имею возможность сегодня сказать слова на открытии конференции, которая уже проходит не первый раз и зарекомендовала себя как очень качественное, а главное нужное научное мероприятие в рамках развития.
And we have it contributes to the development of pharmaceutical industry in Russia, and I would like to express the gratitude for the interest to our conference, to all those connected to us, and we have more than 600 connections today. We believe that this number will grow in the course of the day. Dear colleagues, we know that the pharmaceutical branch industry in Russia enjoys very intense attention, for priority attention by the government of Russia, and we know there are a lot of advances in medicine and pharmacology, and there is a great effort to focus on breakthrough in oncology and in combating the cardiovascular diseases. And we are in the focus of this kind of developments, and Russian People's of Friendship University pays great attention to these areas of advances in science. And in the coming decade, the pharmaceutical industry and the healthcare system in Russia in general, it has a big influx of investments from the federal budget, and some projects at our university are also funded in a centralized way, and there will be a new unit established at the university cluster of the innovative medicine, and several laboratories will be established and here, and it will be the backbone of our activities, and combine the activities of different kinetic biological laboratories. And we are very hopeful that our efforts and our endeavors will serve the main idea of promoting the Russian science and pharmacology in development of new pharmaceutical substances and improving their qualities, new, and it's very inspiring that we are actually in the focus of all those developments, and we train specialists who will contribute greatly to the advances in this field. I wish fruitful work for the participants of the conference, which will contribute to the development of pharmaceutical research, and I believe that the time you spend listening to the presentations will be very efficient and instructive, and add your competence and knowledge which can be applied in your work. Thank you. Many thanks, Andrei Kostin, and we are very hopeful that today we will be discussing at length on interaction of business and higher learning institutions for advances in new in pharmaceutical area, and you have not disclosed all our plans which we have for the expansion of our research unit at the university, but we will share it at the next conference. And now I will give the floor to Philippe Romanov, who is Director of Department of the State Regulation of the pharmaceuticals in the Russian Federation. Philippe Alexandrovich, do you hear me? Oh, good morning. Yes, you're loud and clear. Thank you very much, Philippe, for having the interest in our conference. It's a traditional event that you participate in the conference. We have 620 participants of the conference, a lot of universities and foreign participants listening to you. The floor is yours, dear colleagues. Thank you, Rimma. I'm very happy to welcome everybody at the International Conference of Harmonization of Approaches to Pharmaceutical Development. I would like to thank Andrei Kostin and Rim Abramovich for their contribution in arranging this conference. And we are working on the access of drugs produced abroad to the Russian market, and we have novelties in the regulations and new degrees that the government of Russian Federation released quite recently, 444 decree on the handling of pharmaceutical substances, especially related to the COVID-19 infection. And this kind of new regulations had a great impact for the internal and foreign market. And now we have transited to the electronic submission of applications and all relevant documents for registration of medicines. And we are working on lifting the barriers on movement of drugs across the country and across borders. And what is the current status? As you know, in the Russian Federation, we were first to register a vaccine from the Gamaleya Institute, anti-COVID-19 vaccine. And yesterday we have a decree on extending the specific nature of regulations of medicines related to the COVID-19 epidemic. And we now have a supremacy of the law of Eurasian community, Eurasian unity, and it augments the law of 61. And Russia is the leader on decisions taken within the Eurasian Union, and we have registered received application for about 400 medicines, and quite recently we issued 62 new licenses for the different kind of medicines and other medical products. But it is our colleagues at all across the pharmaceutical industries know that we have harmonized the requirements for application submission for medicines, and in a number of cases we require additional trials. And I believe that the producers know about these novelties, and we have a special period which will last only until the end of this year, and starting the new year, all registration procedures will be in line with the law adopted by the Eurasian community. And once again, I would like to wish you every success, and I believe that this conference will be a good tool for advancement in the use of new pharmaceutical substances and medicines. Thank you very much, Philip Alexandrovich. And now I would like to give the floor our traditional partner, Professor Abed Chasse from Smith Institute of Plant Science and Genetics from Hebrew University of Jerusalem. And I would like to give him the floor. And we know that he always have very exciting presentations. And I believe all questions shall be sent through chat or email. Thank you. Adet, we welcome you. Can you hear us?
Professor Шасеев расскажет вам.
Professor Shaseev will speak about a very interesting startup he launched recently called Plant, and probably he will add to some new information on this research. Dr. Shaseev, the floor is yours.
Good morning. Can you hear me?
Да, слышим хорошо, Адет.
Yes, you are loud and clear.
Can you hear me?
You are loud and clear.
Да, слышим и видим. Презентация выведена.
Can you hear me, Professor Abramovich? Can you hear me?
Yes, they hear you, and you can start.
Okay, so I would like to use my presentation, and because in this one, the one they have, there is no connection to the videos. So if they can allow me to share my screen. Grisha, can you tell them to allow me?
Профессор Абрамович, вы можете разрешить ему сделать share screen? Пожалуйста.
Да.
Он тогда со своего компьютера будет показывать.
Хорошо.
Okay, let me see. Here, let me try. Yes. Can you see my screen now? Can you tell me? Can you see my screen?
Yes. Do you see your screen?
Thank you very much. So thank you for the invitation. I'm going to talk about biofarming of plant-derived human collagen, a novel scaffold for regenerative medicine and wound healing. Before I start, I would like to quote the former Minister of Oil of Saudi Arabia, who said something smart. He said that the Stone Age did not end for lack of stone, and the oil age will end long before we run out of oil. And I think for a good reason, because biomaterials are really much better than synthetic materials. Now, that should not be a reason to criticize our scientists for not making good synthetic materials from oil, simply because they didn't have enough time. 200 years of modern science and industrial revolution is not enough time to really generate really good materials, while nature had 3 billion years to develop some of the most amazing materials that we wish we had in our possession. Yet, our medical doctors still glue and screw synthetic implants into our body. And I don't think that this is a great idea. And why is that? Because synthetic materials fail. Many times they fail because they're not strong enough for their performance.
Рима Александровна, по-моему, что-то тут не в порядке.
Да, Гриша.
Rima, we have some technical glitch and we experience interruption from Jerusalem.
Да, уважаемые коллеги.
Well, dear colleagues, it's inevitable glitch, and we believe that the reconnection will be established, and Aded Shisoev will continue his presentation. I just want to add that this project is extremely interesting, and coal plant, the Compal plant company is startup which was built there, is selling products to 45 countries of the world, and they do monoclonal growth of cells, and they are part of among the sponsors of our conference.
Да, уважаемые участники конференции,
Well, dear participants, not only we have this pandemic issue, but we even have some lockdown in connections. But the professor is reconnecting right now, and I believe he will be back with us.
Hello.
Алло, Адет, мы вас слышим хорошо.
That we can hear you well.
Да-да. Григорий, может быть, вы-
Go ahead, please.
Ladies and gentlemen.
Итак,
Да, уважаемые коллеги, сейчас мне как раз звонят из Иерусалимского университета.
Now they are calling me from Jerusalem University. Сейчас, кажется,
Уважаемые коллеги, да, произошла такая накладка.
esteemed colleagues, they have disconnected electricity in Jerusalem, so there is no need to connect with the Professor Shosev. We apologize, and perhaps they would approach us once again. And with this, I'd like to turn it over to our partner, Professor Masuro Yoshida. This is the Division of Metabolomics Research, Gastroenterology, Kobe University Graduate School of Medicine. And I'd like to add that the beginning of metabolic research in our center was actually related with Masuro Yoshida. And when we realized that 10 years ago, he managed to introduce that into the general system of healthcare, and he is specializing in the cancer of bows, and today's conference is dedicated to the memory of Viktor Chestikov. And 3 years ago, he initiated these researches, and we are very thankful to Professor Yoshida that he got connected to us, and we've been organizing this for the 3rd year, and this year he responded, and we started the research, and we are going to brief you on clinical studies, clinical and pre, and Mr. Dmitry Viktorovich Chestikov, the son of Mr. Chestikov, he will make a presentation, and his research institute will be involved of biology by Belazersky, and now the floor is given to Professor Yoshida.
Ladies and gentlemen, thank you for giving me a nice opportunity to present my work. Today, I would like to talk about our metabolomics study, especially focusing on biomarker research. It's today's content. First, I would like to talk about background and present state of metabolomics. And second, I will explain how to measure the metabolite. And finally, I will explain our metabolomics studies, focusing on biomarker discovery. This slide shows omics studies. The word omics means examine everything. Genomics is a large-scale study of genome, such as genome-wide association studies. Proteomics is a large-scale study of proteins. It's also getting popular. So metabolomics as well, it's a systematic study of small molecule metabolites. It is possible by the recent progress of mass spec and analytical software. Why metabolomics? It's a smaller numbers compared to genome, RNA, and proteome. So human genome estimated about 23,000, human functional RNA about 100,000, human proteome about 1 million. In metabolome, human metabolome estimated about 3,000-4,000. So enzyme-related genes are less than 1,100. So metabolites have been well investigated in biochemical field. So we know where glycolysis pathway, TCA cycle, pentose phosphate pathway, etc. Also, metabolite is close to the phenotype. So alternation in genome and proteome do not always change in phenotype due to homeostasis. Metabolite is no species specificity. So analytical methods are available to samples from different species. Also, metabolomics could detect specific metabolites from foods, such as pesticides. Next, I will explain how to measure the metabolite. So metabolites have a great variety of physicochemical properties, shown here. So, therefore, multi-platform system is required. In my laboratory, we set up 4 different kinds of systems shown here. Water-soluble cationic compound, such as organic acid, sugar, sugar alcohol, we utilize GCMS. Fatty acid and lipid, we utilize LCMS with liver phases. Water-soluble anionic compound, such as sugar phosphate nucleotide, we utilize LCMS with ion pair method. Amino acid, we utilize LCMS with PFPP column. So using 4 different kind of systems, we can measure 4 to 600 metabolites from one samples. I will explain how to measure the metabolite by GCMS. So GCMS have a capillary column for metabolite separation in the column oven. This column oven temperature could increase from 100 to 320, and metabolites are efficiently separated at their own specific boiling points. And each metabolite is fragmented by a 70 thermoelectron, and also observed EI spectrum is compared with database spectrum. This is one example of TIC chromatograph obtained by GCMS of serum. This comes from the pancreatic cancer patient, and this comes from the healthy volunteer. When we overlapped, you can see some metabolite is changed. So our collaborator made a software which can identify the metabolite and semi-quantificate it automatically. This is the metabolite databases for identification by GCMS. Blue one, alanine, glycine, serine, it's a group of amino acids. Light blue, boric acid, lactic acid, it's other organic acid. Yellow one, lauric acid and margaric acid, it's a fatty acid. Others, including alcohol, ketone, nucleotide, could be identified by GCMS. So, next I will show a metabolic studies for biomarker discovery. This slide shows each cancer mortality rate in Japan. Left panel is a male and right panel is a female. Focus on colorectal cancer. You can see colorectal cancer is the third common cancer in male and the most common in female. The number of colorectal cancer patients have been increased with a Western-style diet. Briefly explain about colorectal cancer. So early colorectal cancer is resectable, and completion remission rate is almost 100%. It's good. To detect CRC, we utilize occult blood tests, but false negative is a big problem. Also, conventional tumor markers, CEA or CA99, is a lower sensitivity, especially at the early stages. Imaging methods such as CT or MRI is not applicable to very early screening. Colonoscopy is an invasive procedure. When CRC is first diagnosed, 40 to 60% are already advanced. It's miserable. Therefore, good screening methods are needed. To establish a method, first, for the training set, we enroll 60 patients and 60 healthy volunteers, respectively. There are no significant difference in age, sex, or BMI. Each stage has 12 people, and the purpose is to detect at least stage 2, which colorectal cancer could be removed. Serum Metabolomics by GCMS. As a training set, a total of 131 metabolites was identified in 50 microliters of serum. As a first screening, we confirmed of the metabolite as below. Not derived from serum, stability through the analysis, intra and inter-day validations, and increased or decreased in colorectal cancer patients. And finally, 27 candidates was selected. There are selected metabolites by first screen. From top to bottom, increase the metabolite to decrease the metabolite in colorectal cancer patient. We exclude metabolite from foods and selected top 10 metabolites. As a second screening, we utilize stepwise selection, and then we constructed logistic regression model. I will explain in the next slide. Stepwise selection is a method that selects metabolite objectively from candidates. Multivariate linear regression model. It's a prediction of y using variables shown here. x1, x2, x3, x4 is a variable. So how we can predict the diagnosis using variables? Set dummy healthy 0, disease 1, and output of the prediction model need to be converged within 0 to 1. So this is the formula of multivariate logistic regression model. And p-value, in this case cutoff value, is determined by ROC analysis. So, as a stepwise selection, 4 metabolites are selected: 2-hydroxybutyrate, aspartic acid, and quinoline and cysteamine. It's a coefficient A, B, C, D. And by ROC analysis, AUC is 0.9097. And sensitivity and specificity is both 85%. For the validation of prediction model, we collect new patients around 60 and measure the metabolite from these people and applied them to our prediction model we established in previous slide. This is the result. Top panels come from the training set and bottom panels come from the validation set. So, as you can see here, at stage 0 to 2 early stages, predictive model is a high sensitivity compared to the CEA or CA99. That's a short summary. We constructed stepwise MLR model based on the result of training set between healthy and colorectal cancer patients. The calculated prediction model with training set had a good performance. When applied to validation set, the predictability was maintained. This is a selected metabolite and this is a formula. This method can also be applied to pancreatic cancer. These are selected metabolites by stepwise selection and also prediction model for pancreatic cancer detection. It works well as well. Currently, we promote full automation of whole process from extraction of metabolite measured by mass spec to diagnosis prediction. This picture is an old metabolite extraction machine from plasma. It's a prototype, but works well. We also utilize GC triple quad mass spec for high productivity and high performance. We also prepare more than 15 of stable isotope labeled metabolites shown here for quantitative measurement. Percent RSD y determined by internal standard correction, in this case shown here, and also by stable isotope labeled metabolite correction shown here. Compared with these, you can see a stable isotope labeled metabolite correction made percent RSD smaller than internal standard correction did. We also try to take measurement time shorten by using two different kinds of columns. In this case, it takes only 3 minutes to measure the 9 metabolite. We also make our prediction model better by using more patients, especially early stages, and volunteers. In this case, around 300. We established a new prediction model using 8 metabolite. And as you can see here, this model have a high sensitivity and high specificity compared with CEA or CA99. Currently, we are working prospective study of this prediction model. So next question is, can metabolomics predict therapeutic and side effect or not? So this is a schema of biomarker research for the therapeutic efficacy prediction. 26 esophageal cancer patients at stage 2 to 3 are enrolled, and serum sample was collected before neoadjuvant chemoradiotherapy. And then these patients underwent 2 courses of infusion of 5-FU and CTDP, plus concurrent irradiation. And then surgical resection was carried out 6 to 8 weeks after the completion of neoadjuvant chemoradiotherapy. Let's see the result of histological response. By surgical resection, we found out of 26, 13 people is a complete remission, so tumor is gone. And 13 people is a non-complete resection, still tumor is there. So these two groups, age, sex, tumor location, clinical stage, have no difference. Before any treatment, serum sample was collected from esophageal cancer patient, and then serum metabolites were analyzed by non-target metabolome analysis with GCMS and LCMS. And then identified metabolite was measured by a targeted metabolome analysis. So we examined what kind of metabolite were changed between CR and non-CR groups by non-target metabolome analysis. In result, several metabolites including serine, arginine, glycine, aspartate, betaine, and arabitol were identified. So next, we prepared stable isotope labeled metabolite respectively and performed quantitative analysis, just shown here. So as you can see here, serum, serine, arginine, glycine, arabitol significantly changed between CR and non-CR groups. Let's see the result of Cox proportional hazard model analysis. So variable is the age, clinical stage, and 4 metabolite, serine, arabitol, arginine, and glycine that we identified before studies. So univariate analysis revealed only serum serine was significantly correlated with time to progression. This is a long-term prognosis. Left panel is time to progression, and right panel is cause-specific survival. On the basis of highest sensitivity and specificity of the ROC curve of prediction of histological response by serine, cutoff value 110.6 micromolar was obtained. You can see here high value of serum serine before treatment lead to the bad prognosis. In other words, the value of serum serine before any treatment is predictive marker of neoadjuvant chemoradiotherapy with esophageal cancer patient. This is a conclusion. Metabolomics could provide a useful information in the medical field, especially for discovery of disease biomarker, and we have no time today to show, but still good for the finding of novel therapeutic agents. Thank you for your kind attention, and also I thank all my collaborators, especially Dr. Shin Nishiomi at Hyogo Medical College. Thank you for your attention.
Огромное спасибо, профессор Йошида. Но я хотела бы вам задать...
Professor Yoshida, many thanks. Can you hear me?
Первый вопрос.
And my first question is, I would like to ask some questions, and my first question will be as follows.
Первый вопрос я хотела бы, чтобы вы с нами поделились, а сколько
Please.
вам все-таки удалось предотвратить смертей от рака, да?
Can you share with us how many deaths you succeeded to prevent by introducing your methods? And my second question is, have you actually made your research only on Japanese nationals? And we wonder if we can also apply similar research here in Russia. And we have also a breast cancer research and this colorectal cancer research. And we wonder if you can come, once the epidemic is over, you can come to our university and we can work out with you the design of a new research in this field.
Доктор Йошида, мы вас слышим.
Hello, can you hear me? Can you hear me?
Да.
Yeah, no interpreter. I cannot understand the Russian.
Dr. Yoshida, you have to
yes,
the translation. It's on bottom of your screen, and you have to choose an English.
Bottom of screen.
Yes, in Zoom.
Yeah, I use the Zoom. Yeah.
Yes, and you have to.
Okay, got it.
Okay, can you hear the interpreter, please? Can you hear the interpreter? Can you hear the interpreter? Can you hear the English translation? Can you? Just wave your hand if you can hear English translation. My question is, how many cases of death you managed to prevent? And how can you, did you implement it in your practice? Can you hear the translation?
Sorry, I cannot translate it. Hmm? Hmm?
Professor Yoshida, do you hear us?
Can you hear English translation, Professor Yoshida?
Hmm? No.
Professor Yoshida,
Only Russian.
only Russian?
Hmm.
Oh, in this case, I think that we have to ask in English. Dr. Yoshida,
Yeah.
we have several questions. For example,
Okay. Okay. Come on.
okay, thank you very much for the connection to our conference. And we really there was simple. I think that changes in the program of the conference in the plenary session because the previous speaker has some problems.
Yeah. Okay.
Yeah,
Okay. I
yes,
understand.
and that is why we started your presentation a little
No
bit
problem.
early. And Rima Alexandrovna Abramovich, director of Shared Research and Educational Center of Ruden University, has some questions. And the first question is, how many lives does or this did this method, of early diagnosis of oncological diseases saves in Japan?
So it's very difficult question. We are just trying. We need a prospective studies.
And and the second question, did this methodology used in Japan? Is it possible? And the third question, is it possible to use it in, for example, in Russia?
Yeah, of course, because in the we use a Shimazu Corporation machine. So and also in the Shimazu have some branch in Russia in Moscow. So you can ask, and we will teach you. No problem.
And Rima Alexandrovna Abramovich wants to invite you to our university when the pandemic of COVID-19 will end and the borders will be opened. And and in our university, in our center, we have a laboratory which is equipped with Shimadzu equipment. And in this case, we are ready for example, for mutual for mutual, for example, experience in the developing of maybe new methods of early diagnosis of different socially available
that's right.
diseases.
That's interesting. Nice idea. I think I'm very happy to collaboration with you.
Yeah, because we have such equipment like LCMS with triple quad Shimadzu production, and then we have Shimadzu gas chromatography with monoquad, And also we have different other chromatographies produced produced by Shimadzu, and that is why I think that it will be really interesting.
Yeah, I'm very happy.
And also we have a lipidomic program for lipidomics and
Yes.
for oxylipin detection and quantification.
Yeah, actually, we also have a LCMS system using the lipidomics studies. And also I can share our information with you.
Thank you very much, Dr. Yoshida, and we are very happy that you find to connect our conference because it was really nice to hear your presentation. It was very, very interesting. And I think that Rima Alexandrovna Abramovich also wants to say some words to thank you.
Thank you very much, Professor Yoshida.
Thank you.
We await you in our university in the
Yeah,
summer
thank
and
you very
spring
much.
when you're convenient for you.
I'm waiting.
When you convenient for you, when you finish COVID pandemic. Now we good collaboration with Surgutsky Medical University in Rязань, and the Surgutsky Medical University won't study the cancer in the beginning because have very big center and need for our collaboration. Thank you very much.
Thank you very much.
Thank you very much. Goodbye. I think that we will.
Итак,
Thank you, Dr. Yoshida.
Thank you.
Да, уважаемые коллеги.
okay, esteemed colleagues, and our Israeli colleagues wrote us that they have shut down of electricity, and we cannot continue transmission from Jerusalem. Therefore, we will just upload this presentation on the site of the conference, and later on you can send the questions to us, and we will do our best to make our Israeli colleagues to respond to your questions. And now, the colleagues, I would like to talk about our center. My presentation is being downloaded. Dear colleagues, today's conference have collected for the third time the specialists in pharmaceutical industry, both domestic ones and from abroad.
Наших постоянных участников получилось, что наша
1/3 of our regular audience, 1/3 of our participants have been traditionally connected to us, and we have more and more members. And the colleagues, we cannot see the presentation in Zoom. We have been discussing pharmaceutical development at length. Today we will have several sections, and our regulators, the research institutions will dedicate their presentation to the quality of pharmaceutical substances and the development of them. And today, it's our first time we arranged the conference online. And again, I would like to introduce what kind of integration we have here in the science research and the educational institutions. And after lunch, in more detail, you will learn this Alexey Viktorovich will present his interdisciplinary review where he will demonstrate the current status of research jointly with the Russian Academy of Sciences and the Basel researchers in the field of microbiotic resistance and anti-tumor medicines. And the Institute of Dimar Rogachev Institute will also join us and talk about it. Deputy head of Department of the Regulation of Medicines from Ministry of Health of the Russian Federation. He'll speak about the risks of the use of medicine, especially it concerns the pediatric dosages and the pediatric application of medicines, and here see a great shortage of the products, and there will be a specific presentation on it, and we will also talk about harmonization of standards in this field. And I know that another interesting topic is transbuccal films, Medazolam program, and epilepsy issue in children. And now there are certain advances in medicines there. Therefore, both our research, business, regulatory bodies, and the high learning institutions in a concerted way will discuss our collaboration or when next year within this framework. Alexey Viktorovich Kitilyan, Garyanov Sergey also will talk about it on the clinical and preclinical trials with the son of Dr. Chestikov. They will speak about metabolomics and speak about the latest advances, and we have acquired the latest equipment, and we believe that in 3 years we have done a lot. More than 25 articles were published in different magazine journals, and we probably can on our site put a link. And today our audience numbers around 60 different high learning institutions, 35 different research centers, around 160 connections of our pharmaceutical industry representatives. We will just talk about how we can help our pharmaceutical companies to improve the access to international market. What projects we can do jointly. And last year our conference was attended by 880 participants, and we have another section which was related to metabolomics of herbs and the wine production. But this time we dedicate this conference to late Professor Viktor Chestikov. Unfortunately, he died in February 2020. But his cause, his research he started in pharmacogenetics and metabolomics is going on. He was the pioneer of personalized medicine, and I believe that the pharmaceutical producers for whom he was developing new molecules will recall him and pay tribute to this man, and his cause is still being promoted and advanced, and jointly with our diagnostic center, with our medical institute, we continue research with jointly with Bizel-Ozerski Institute, where Victor, his son, is working. And I would like to thank Professor Zырянов from Hospital 24 of Moscow. And there we address issues not only of socially significant diseases and oncology. And there we prepared an article to be published by the end of the year on early diagnostics of breast cancer. In Hospital 24, we started a research related to isolation and development of isolipid panel for diabetes, ischemic heart conditions, and again, I would like to thank our long-time collaboration with Moscow Research Institute named after Herzen, this oncologic center. And I believe the colleagues are connected to us. They have collected a great variety of samples for us. We have analyzed them. We would like to thank the clinical center where the standards have been collected to use as reference for the diagnosing breast cancer. And by the end of the year, I believe we will have around 300 samples of them, and we have, and more detail will be presented in the report by Sergey Vladimirovich. And therefore, what Viktor Chistikov has started, and his endeavor never ends. And our center has been established 11 years ago. And within this year, we went to making the entire range of research on molecules and including the herbs. And we have GMP unit where we design innovative molecules. We have a vivarium for preclinical studies, as Andrei Kostin said today. Next year, this strategy of Ruda development until 2020, it's for the decade, and it will be approved soon. And this plan includes to build a cluster of laboratories, and it will be an international level vivarium, and this research in oncological field can be jointly carried out there. And we can do both the development of new molecules, synthesize them. We can also attest the reference samples and the pre and final treatment. And we have patents for transbuccal film production and horn inhalers and transhormonal plasters. And therefore, we have the experience of implementation of orphan drugs like neogrilite. We believe that we can also be of help if somebody wants to produce orphan drugs. Here, I would like to show you the focus of our development in personalized medicine. It's what Dr. Chistikov once did, and he was passionate about. We do it with the Basel University, and each person has its own biogenetic background, and the Titachrome 450 is individual for everybody. Therefore, jointly with Basel University, we developed a system where we determine personal pharmacotherapy. I would like to thank our colleagues now. And we heard the Japanese professor, Professor Ishida, report about metabolomics for early diagnostics of oncological diseases, and we do a similar research on the metabolites and low molecular weight intermediates or final products of biochemical reactions. We have been doing this study for the last 3 years, and I invited the Professor Ishida to Moscow, and we believe that we will continue this research on lung cancer and stomach cancer and breast cancer, and this is the range of the research in our laboratory. It's metabolomics. This for the last 3 years, and another one they're collecting, building up a biobank, which is needed to work with different types of diseases, and we will have Johan Hulliver will speak about preclinical studies, and it will be a very interesting presentation on opioid medicines and how to combat drug abuse, and he'll speak about this abusive deterrent medicines and or which concern the modern and perspective medicines. Um, Now I would like to share you what kind of test systems we have to assess activities of 400 P450 cytochromes, and it is in the clean areas, and we perfected honed the technology of doing operating this equipment. We are doing research on animals, and we are coming to presenting clinical samples, and uh, next year we are going to start clinical samples, and Basel University sent a question on sending these test systems to their hospital, and I believe this is the link with practice. And what else I would like to share with you, as our conference is dedicated to harmonization of requirements to the uh, pharmaceutical developments and this diffusion of pharmaceutical substances, and this is an innovation topic for us, and we've been doing it for the last 3 years, the hot metal intrusion. And this method is used when the molecule doesn't dissolves or distributes well, and all our research is focused on the increasing bio accessibility for bio medicines which do not dissolve and digest it. Therefore, we have designed the anti-HIV drug, which uses some polymer and active pharmaceutical substance at increased temperature, and the range of temperature is being very difficult issue, because we did more than 100, even 150 tests to find this good melting temperature where polymer is mixed with active pharmaceutical substance and destroys it crystallized structure and creates a hard dispersion. And it leads why we use this hot melting substance is to have a prolonged effect for the medicine, and or a regulated release of active substances, and when we need to reduce adverse effects of toxic substances and increase the bioaccess and bioavailability of it. And jointly with Basel University, we have selected a polymer and technological conditions and the procedures. We started to manufacture GDP type capsules, and we've got quite promising results in on 36 volunteers, and we have proven better accessibility, applicability of hard dispersion, and our substance become more soluble, and it digests better, and we managed also to reduce the dosage and adverse effects of this medicine. And another research which is currently underway on transbuccal films and transdermal patches. For 10 years, we've been doing different of more than 250 projects, and 10 state orders were fulfilled. That's time to finalize my presentation, and again I would like to underscore that at this venue we combine the efforts to introduce science into practice and create consortia for the innovations, and train new specialists, the cadres which will be implementing these projects. And we have a specific program of 40% of which is dedicated to the promotion on pharmaceutical technology. And again, I would like to inform the high learning institutions that this year we established a dissertation council on the technology of manufacturing medicines. We are looking for your postgraduate students which can publish their articles with us, and they can join us in the research. We provide quite good equipment for such research, and maybe we can do some metabolome research there with using mass spectrometry, which is quite a promising approaches. And our postgraduate department, jointly with the Basel University, started a number of efforts, and I would like to thank the Professor Julio on providing us all necessary documentation on the on research, and they are ready to invite our students to their site. Boris Konstantinovich, you are connected. I would like to give the floor to Professor Doctor of Medical Science, Dr. Romanov, Boris Romanov, Deputy Head on Science of the Center of the Medical Expertise.
We have big problems in developing pharmaceuticals for kids, and we are also involved in this work. And we know that GILS also declares and develops the platforms for formulations. We would like to hear what kind of problems we are facing in this respect. The floor is yours. Thank you very much, esteemed Rima Alexandrovich. I thank you for invitation to take part in this conference of RUDE-N. And this is the institution, and I worked on biochemistry project 15 years ago. This organization is very familiar to me. And then I started to deeply study pharmacology, and for 10 years, I've been involved in safety of drugs, and we are working with the organization which stands close to the Department of Pharmaceutical Regulations. And Philip Alexandrovich made a bright presentation in this. Allow me to switch the presentation. The topic of my presentations are the risks of use of formulations among children, and what kind of risks could be visualized concerning the pharmaceuticals as per the regulatory documents of Eurasian Economic Union. The decision 87 was adopted in 26. Any types of risks related to quality, safety, or efficiency of pharmaceutical formulations towards the health of patients or populations, or leading to unfavorable and hazardous effect on environment. Of course, concerning the kids, it is very critical factor in this field. The safety of pharmaceuticals and efficiency and accessibility are the basic consumer properties of pharmaceuticals, and they are in turn derivatives of the quality of the formulation. If the quality is satisfactory or good, other characteristics would be derivative. And the quality of drug is placed at the stage of different processing and developing, be that pharmaceutical development followed by pharmaceutical development, passing over to preclinical development, in vitro, in vivo, experimental models, animals, and then in turn gives the prerequisites for further development and clinical testing, including those formulations for pediatric purposes. And all these preclinical, clinical, they are part of the registration dossier, which defines the requirements of the quality of pharmaceuticals, describing its accessibility, safety, and efficiency. Receipt of actual data about the risks. It represents an activity which is called as pharmaceutical supervision, consisting of pharmacon and Roman word vigilaria. And we get the term pharmacovigilance, which is pharmacovigilance. That's the type of activity studying problems related to the safety of pharmaceuticals at all stages, starting with the development, ending with the broad circulation of pharmaceuticals on the market. Since 1966, the World Health Organization is supervising the safety of pharmaceuticals, which developed the international program. And here is the slide with the countries representing and taking part in this activity and program. Russia is also represented there. And the program consists on the collecting of information, so-called SAOMS forms, the primary reports about cases of unfavorable conditions or inefficiency of certain formulations, and the development of recommendations. Global pharma vigilance is represented by a gigantic amount of data, and this is the database of Visualize contained more than 23 million cases of individual messages about unfavorable reactions on pharmaceuticals, including more than 1,900,000 unfavorable reactions towards pharmaceuticals among children at different ages. And what are the basic indications of the work of the system for kids? This slide demonstrates top 10 pharmaceuticals during the whole period of the work of this system, and this apparatus initiated its work since 1968. And you can see that top 10 formulations which were indicated in this database, they are represented by vaccines. And this is clause number 9. This is paracetamol, a synthetic substance. Why do we have so many vaccines? This is because of the broad scale of use of vaccines and represents scalable use of those drugs among kids, and the tendency, the number of messages on unfavorable use of vaccines is diminishing, and more information we get about the influence of synthetic formulations. If we analyze the year of 2020, it started very very difficult in the beginning about the unfavorable situations, and that was related to the distractions of medical personnel to fight pandemic. However, the number of indications and messages as of the 1st of January towards the 12th of November, the international monitoring system received only 2,000 messages, including the problems with formulations, 7% approximately. And what are the pharmaceuticals were mentioned most often reported as a subject that certain unfavorable conditions emerged. And a very interesting fact, the first two formulations, as per reports of 22, this is Adepolan, and this is to treat the eruptions. And actually, young people are taking care of their appearance. They use different pharmaceuticals with having active ingredients. And the first actually problem amounted to Adepolan when a kid is using certain formulation taken from his parents, or he's trying to use something by his own understanding. And actually, this are basically cosmetic things are used in dermatology, almost 15%, and a fairly a big number of messages coming on the harmful effects of vaccines. And the first place is taken for meningococcal vaccines, pneumococcal, then polyvaccine, sodium chloride, and others. Somewhere in the middle of this list, unfavorable reactions is also attributed to salicylic acid. And in order to soften the stiff parts of the skin. And also unfavorable reactions are being reported. What happens in Russian Federation? On the slide, you can see analytical data as per the unfavorable reactions towards the drug. And I would like to mention that Russian Federation is very well represented, and we are the country, are the full member of the World Health Organization, and we report information of very high quality, and perhaps some countries may omit certain important information, forgetting to mention the age or whatever. So Russian Federation is very accurate, and only the messages of highest quality, perhaps 95 scores out of 100 is the characteristic of the quality of messages. And what are the most often cases and mortal cases in the case of use of certain drugs? So these are about 12% out of all reports and messages, and international non-patented names are represented, and titles are represented here. First of all, lidocaine, ceftriaxone, cefotaxime. This is for anesthesia, and aspirin and its derivatives, and paracetamol. So these are the formulations most often reasons for lethal cases and mortal cases. And the Russian Federation actually has adopted the same system of step-by-step registration of pharmaceuticals. And first, this drug is developed for grown-ups, for adults. Once it proves to be safe for the adults, so they continue to develop it for children, for early ages, for 12 years and further on. That helps us to prevent a large number of adverse reactions, and we get a fairly safe drug in the end, having been properly tested for broad use. What do we have to do, and what do developers of formulations should do to satisfy all the requirements towards the reporting of safety of formulations? As of 2019, Roszdravnadzor has launched the second version of its internal information resource, which is open for registered users. This is the database of Roszdravnadzor. And if you are a developer or working with the drug, you need to get registered in this resource, send a message to this site, and you will get a login and a password so that you could enter information in this database. And this is the state-of-the-art database, matching all the necessary platforms, and if it has certain solutions for processing, so they are linked with the international platforms, and this information could be sent automatically. So you would comply to the requirements offered by the developer in terms of regulatory requirements. I thank you for your attention. I'm prepared to take your questions, if any. Thank you very much, Boris Konstantinovich, for your presentations. If there are any questions, please forward them to the site of the conference, and Boris Konstantinovich will respond.
And Elena Leonardovna Kovaleva is next to speak. She is Doctor of Medical Science. She is the head of the Scientific Center for Expert Assessments of Medicine Application of the Ministry of Health of the Russian Federation. And her presentation is called The Current Issues of Expert Assessments of Quality of Pharmaceutical Substances. Dear colleagues, good afternoon. My current presentation not only will dwell on the expert assessment of medicines, but we will also speak about standardization of drugs. Although the registration procedure of medicines follows the procedure of Eurasian Economic Community rules, but it is just picking up speed, and we are still using the national registration procedure, but still we observe an quality leap in our operation because we have already established the regulatory basis for quality assessment of medicines marketed in the Eurasian Economic Community. And they all are in line with the WHO, ICH, and EMA. On this slide, I refer to the basic documents which were approved recently on the quality of medicines and medical products. And my presentation will be at the site of the Eurasian Economic Committee. Some documents are mandatory, others are just guidelines, and among those recommendations are assessment of DNK-reactive substances, inhalers, nasal drugs. And I would like to draw attention to two guidelines, which for us, the experts in the Russian Federation, are novel ones, because they are stipulating the requirements for compound medical molecules for those which are used for we are not just assuring quality, but we see examine the quality and quality of coating, and with medicines based on the colloid ion, and these parameters first are studied and they're included in so-called specification list, and we determine their stability, defined the critical values of these qualities, and the document specifies what kind of values of quality should be reflected in the application package. And once I will not read all these parameters, they are not, this is not an exhaustive list, but still this guidelines is in effect. And on the 11th of August 2020, we have a historical step on approving the Order 100 by Collegium of Qualification Committee on approving the first volume of Eurasian Economic Union Pharmacopoeia, and it is a book, but it looks like a book, but unfortunately this kind of document is only accessible as a set of critical articles and requirements, and just in the first volume we declare that all the applicants and the experts can still have a better picture about what this volume includes. Among the Eurasian community, only 3 countries joined it, and the Republic of Belarus and Kazakhstan. They operate in line with the European pharmacopoeia, and Russian pharmacopoeia is not oriented on one foreign leading pharmacopoeia, and we do it with due regard to our national specificity and with due regard to the leading international requirements, and adopt in the conception of harmonization of pharmacopoeias of states that we shall harmonize the basic European pharmacopoeia and as the level of the first tier pharmacopoeia, and then the US pharmacopoeia is the pharmacopoeia of the second level. And we harmonize the terms, the measures of units of measure, and requirements, acceptability standards, and requirements to quality standards for drugs and medical products. Once we don't have a common pharmacopoeial articles, that still we can refer to the national pharmacopoeias of the Eurasian Commonwealth members, and if this state doesn't have their own pharmacopoeia, they can use pharmacopoeias of other members. And we had a number of situations when we provided our expert assessment, because sometimes a drug comes to a reference country and they refer only to pharmacopoeia of this reference state. And then they act following the agreement on mutual recognition of medicines, and therefore we decided that there should be at least 3 different pharmacopoeias referred to. At this slide, you can see the content of the first part of the first volume, which includes 157 of general pharmacopoeia articles. It includes the analysis methods and pharmacological tests and the quality assessment, quantity assessment methods. pharmaceutical and technological tests. And current pharmacopoeia has a number of specific features which reflect the current scientific progress, and these distinguishing features definitely affect the preparation of the registration dossier for specific medicines. And the quality standards are very dynamic in development, and more complex methods of pharmacopoeia examinations are used, and they're sometimes mandatory, sometimes they are just recommended, and exclusion or replacement of biological studies by uh, studies on animals, and the inclusion of different methods and norms on solution and disintegration of the drugs are also reflected, and pharmacopoeias in the US and Britain are regularly updated. And our last edition dates back to 2016, and let's look how it differs and develops continuously. We deal with the new quality assessment of drugs, and in according to the latest documents approved by the Commonwealth of Eurasian countries, and there we evaluate the UF range in our analysis. It is identification for the time of the sustainability, and but we do spectroscopy, and two chromatographic methods are recommended to be used, which are based on two different principles, and for quantity assessment, they used a specific method which allows to use the stability of the drug. And we are not talking about opticometric methods of examination, but we deal the chromatography of the drug, and the size of particles. Laser diffraction allows you to evaluate the medium size of particles, and but also the distribution of these particles should be examined, because light dispersion of the particles allows better absorption, and current the analytical methods allow us to evaluate and to measure this method. And what about heavy metals? There is a special article dedicated to it, which also is a translation from the European Pharmacopoeia, and we define this natrium sulfide methods to define the contents of heavy metals, but we also included another article where we deals with the heavy metals, and they include the latest methods to isolate these substances, and we also apply the risk assessment methods for the content of this kind of substances, and they are not only the value of a particular metal is evaluated, but all the impact in the compound and the element additives is an article which governs this kind of requirements, but this is not yet included in the latest edition of Pharmacopoeia for the Eurasian Union. But here I list the number of examinations which are carried out with the use of specific equipment. Hematoxic, mutagenic, additives in impurities in medicines can even in insignificant amount have a big impact on DNA and initiate some genetic mutations or chromosome breakages or restructuring of chromosomes. Therefore, the applicants, when submitting the application, shall provide information on the modern instrumental analysis methods like mass spectrometry, because these impurities, even in meager dosages, can have significant consequences. And we see the wave of the negative information of this kind of impurities are cancerogenic, and or sometimes the drugs which were commonly used then become the criticized, and then the same happens in the European Pharmacopoeia, where there is an article on defining genotoxic impurities for benzol ethers, and the technology of these active substances contains risk of such adverse impurities. The leading pharmacopoeias, apart from general articles, which are mandatory for or include requirements for drugs, but there are also articles which are providing recommendations or informing the producers about certain issues which they will be aware of. And this is new for us, because our pharmacopoeia doesn't include articles of such nature, but we are quite carefully examining what our colleagues doing, and we try to have a similar approach when structuring our own pharmacopoeia, and including this harmonization and or mutually agreeable methods is a trend which describes the current status in this field. And quite recently, test activation of monocytes test was recommended, and we included it into the Russian pharmacopoeia, but it is not yet included in the Eurasian pharmacopoeia. But it is a test for pyrogenic type of substances, and for semi-synthetic or classical antibiotics which received by microbiological synthesis. This kind of tests are not alternative, because LAL test is very specific to endotoxins of gram-negative bacterium, and its sensitivity is much more higher than the in vivo tests on rabbits. Therefore, not only this test activation for monocytes allows us to give up tests on rabbits. But so far, from what we know, the European pharmacopoeias don't include the MET test, although they have the general article on this. As for the alternative methods application, the Russian uh, pharmacopoeia says that it should be subject of validation. And we believe that the sensitivity of such alternative test methods should be comparable with official approaches, and not always a drug can be controlled or pharmaceutical substance can be uh, checked for impurities or which are listed in pharmacopoeia, because the selection of additional substances, antioxidant selection, can lead to the situation when this method cannot be applicable to a particular drug. In European pharmacopoeia, if we talk about the articles, the alternative methods, as a rule, are used for identification of pharmaceutical substances. What about the verification of pharmacopoeia methods? Not always these pharmacopoeia methods can be recreated for a particular drug. We can verify this medicine to prove it's good by and but sometimes the standard approaches do not match, and because of the composition of the auxiliary substances which are included in this particular drug. But pharmacopoeia needs to specify these permissions for alternative methods to be applied. It was quite surprising when several years ago, when the US pharmacopoeia includes more than 10 methods of solubility of a drug, and they just say that the label of the drug should just specify what kind of methods for solubility was used. But this kind of tests include not only methods, not always this method is good, but bioequality of bioequivalence of these medicines in order to launch this drug in the market should be subject of proof. And therefore, different methods can be used to get this result, which have to do something with the synthesis method and the impurities profile can determine the selection of such a method. And quite recently, we did an expert assessment of a new drug when with pharmaceutical substance from two different producers were used two different separation methods. But still, the European pharmacopoeia believes that both drugs meet all the requirements of the European pharmacopoeia. Therefore, currently, we are living in an unusual situation. Previously, we had just one pharmacopoeia article which concerned all producers of a particular drug in Russia. But then the main provisions for such standards were changed in 2001. And then the producers could define themselves what kind of methods they will be used, and the European producers were just using the European pharmacopoeias And the US pharmacopoeia give us an example that methods to check the active medical substance shall be checked with the use of methods applied by the producer. And this is a new vision for us. And so far, it is not included in our pharmacopoeia, but we are carefully studying the current situation in the field. Елена Leonardovna, many thanks for your presentation. Our plenary session is virtually over. My Sakayan is sick. He will not speak today. Maybe his presentation will be then uploaded to the conference site. All questions which you have, especially to Professor Lucida, you can refer to the mail page of the conference or reference site. And dear audience, I would like to thank you for your interest to this international research scientific conference, and I wish you quite fruitful work at the sessions. There will be a lot of reports. All the reports will be published, and there will be 4 sessions, 4 different sections, and some of them will be extremely interesting on the cutting edge of the modern technology. And for the first and another session which will attract a lot of pharmacognosia and herbs drugs, and Marina Lyakina will moderate this session. She is Doctor of Medical Sciences, and the section which is dedicated to Viktor Chistikov will speak about the role of pre and after post-clinical trials. Igor Shokhin will be moderating this session, and I just predict very interesting presentation. And it very intensive work. Therefore, I am just urging you to send your questions to the site, and let's meet in presentia next year. Please send your comments about sessions. As for the format, it will depend on the epidemiological situation, which we hope it will change due to the availability of vaccines, which some even are important already. And we know already how many drugs have underwent the accelerated procedure of approval. And I wish you health and happiness. And this year, normally we cannot do a common picture. Maybe you can send your pictures and we will kind of combine them in a collage, because it was the situation in our country and across the world. Therefore, I'm urging the audience to send the photographs, which we will combine and then upload to our site. I would like to thank you for participation, and see you again.
Трансляция идет. Ну, я хотела бы нашей поддержки технической, да, что мы сейчас конференцию, пленарную сессию.
Well, the plenary session is over. Professor Ben Mueller wanted to speak, but we got in touch with him. He is indisposed, and therefore his presentation will be then downloaded to the conference page in the internet. And as I said, we on some manual on suppositorium jointly with Bern Mueller published, upgraded, and revised. And let's have a lunch. I so pity that you are not with us, because everything is organized here at the university. We have very good cafeteria and a bakery. But I wish you bon appetit. And again, please follow the link and select the section of your interest, and your questions shall be go to a particular page in the internet. And I thank you and bye.